So now I'm going to dive into some of the results from this project, and then Liz will go into how we got to those results using Biodata Catalyst. So before I talk about the microbiome results, I want to define one term. So I'll be talking about alpha diversity. So alpha diversity is a concept that comes from ecology. The gut, of course, is also an ecosystem, just like a smaller scale, and involves microorganisms rather than plants. But some of the same concepts apply. So alpha diversity has two main components: richness and evenness. And this picture on the left is showing an environment that has high alpha diversity. It has both high richness and a high evenness. So the richness is high because there's lots of different types of plants present in this environment. And then the evenness reflects how those plants are distributed across the environment. And so you can see that there's lots of different plants, but the environment isn't dominated by one specific plant. On the right are two types of low alpha diversity. So the upper one has low richness. And so there are not, at least, in terms of living plants, there are not a lot of different types of living plants in that picture. And then on the bottom, we have low evenness. So there's a lot of palm trees, but not a whole lot else, right? The environment is largely dominated by that one type of plant. And just like the picture on the left, like intuitively looks healthier than the two pictures on the right, the same tends to be true of the microbiome in the gut. So higher alpha diversity tends to be associated with better health. And this is not always true across all diseases and everything else, but it does tend to be true across literature. So low alpha diversity has been associated with obesity, diabetes, Crohn's disease, lots of different chronic conditions have been associated with lower alpha diversity. So in this project, we might expect that asthma would be associated with lower alpha diversity. And so we looked at 4 different metrics of alpha diversity that capture, different aspects of evenness and richness. And those are shown in this picture on the y-axis. And then we looked at them in relation to current asthma. And the black on this figure is showing our fully adjusted model. So we're adjusting for all the potential confounders that might obstruct the relationship. And so we can see there's not a strong relationship between alpha diversity and current asthma. Evenness is actually a little bit associated. So lower evenness is associated with slightly higher odds of current asthma, but it's not a very strong relationship. Then we broke down current asthma into non-obese asthma and obese asthma, and we can see a slightly different picture here. So obese asthma and non-asthmatic obesity are associated with lower alpha diversity. And so, as I mentioned, this has been seen, in other studies of obesity, and we definitely see here that the obesity is probably driving this relationship more than asthma necessarily. We don't see the same pattern with non-obese asthma. Interestingly, we actually see kind of the opposite relationship. Higher alpha diversity, higher observed features, so that's a measure of richness, is associated with non-obese asthma. Again, it's not highly significant, but it does cross the threshold of significance. Next, we looked at something called beta diversity, which is comparing the overall taxonomic structure or composition across individuals. And so this is asking whether or not the overall microbiome differs among people with asthma versus people with no history of asthma, or people with asthma and obesity. And so we saw a highly significant difference in the overall microbiome structure between people with asthma and no history of asthma, and then also these individuals with asthma or obesity compared to people with neither. I'm not going to dig in too much into the taxonomic results, but this plot is showing taxa that associated with current asthma on the left, and then asthma and obesity phenotypes on the right. And you can see that there are various taxa that we saw that were associated with these outcomes. I had mentioned that short-chain fatty acids are really potent anti-inflammatory metabolites that are made by the microbiome. And 3 of these taxa that we identify as associated with these outcomes are producers of short-chain fatty acids. And then those are shown in green. And then there's one that we saw that is associated or is often uses short-chain fatty acids. So it's a user of short-chain fatty acids. And that's at the top. And that was associated with increased risk of these conditions. Then we did a causal analysis to see if any of these taxa are causally linked to asthma or obesity. And so we used an approach called Mendelian randomization, which basically uses genetic information in order to get at causal relationships. And so using that analysis, we found that there was one taxa among our results that was associated with increased, or was protective for asthma. That's the pseudoflavinofractor. And then we found one taxa that was protective or causally protective for obesity, and that was this Fornelia. So both of those were short chain fatty acid producers as well. Next, we made a polygenic risk score for asthma, and these plots are just showing the distribution of that polygenic risk score among individuals with asthma versus no history of asthma, and then with these asthma obesity phenotypes. And one interesting thing to note here is that this is an asthma polygenic risk score, so not for obesity, but it's most elevated actually with obese asthma versus, say non-obese asthma. And then when we look at, how well this asthma polygenic risk score predicts our outcomes, we see the same pattern, that it's more predictive of obese asthma compared to non-obese asthma. And so then we wanted to kind of build parsimonious models of these outcomes. And we were wondering, if we have, genetic information, does that improve our ability to differentiate people who have asthma versus not asthma? And, if we have microbiota information, does that additionally improve our ability to differentiate these conditions? And so when we looked at current asthma, we saw that the polygenic risk score improves our our modeling of current asthma above and beyond baseline risk factors. And so that's shown with this p-value here that's highly significant. When we add microbiota features, it again improves the model. So these are kind of adding independent information that's not captured by our baseline risk factors. When we look at the outcome of non-obese asthma, the asthma polygenic risk score actually does not improve the model above baseline risk factors. The microbiota features do. And then for obese asthma, both pieces of information improve our models. And then I'm not going to dig in too much into these results, but I mentioned we also, looked at just the subset of individuals who have asthma or obesity or both, and tried to understand if the microbiome signature of these subtypes differed. And so this plot is showing an ROC curve of our models. And all these pink and red ones up here are the comparisons to non-obese asthma. And so if these lines are kind of closer to this quadrant up here, up in the upper left, that means that the information is more predictive. And so our microbiome information, taxonomic information, showed a distinct microbiome signature for current asthma, non-obese asthma, and obese asthma versus non-asthmatic obesity. So basically, asthma has a distinct microbiome signature compared to obesity without asthma. We also compared non-obese asthma to obese asthma, and that's this black line here. And so this straight black line across the middle is just like random guessing. And so our model isn't that much better than random guessing to differentiate those two conditions. But it's important to keep in mind that we had much smaller sample size. We only had 191 individuals with asthma. And so that comparison had much less power. So some overall conclusions here: gut microbiota diversity, taxa, and functions, which I didn't talk about the functional analyses, but all of those were associated with these asthma and obesity phenotypes. We observed evidence that some of the taxa were causally linked to asthma or obesity. We saw that gut microbiota independently improves models of current asthma, non-obese asthma, and obese asthma beyond baseline risk factors in an asthma polygenic risk score. The asthma polygenic risk score actually associated more strongly with obese asthma than with non-obese asthma. And asthma and non-asthmatic obesity have distinct microbiome signatures. And so we just want to acknowledge all of our collaborators. Robert Kaplan and Robbie Burke are the PIs of this GOLD study, the ancillary study in Seoul. And actually, Robert is one of the PIs of Seoul as well. And then also we want to thank our funding, especially the Biodata Catalyst Fellowship, and two of the Biodata Catalyst help folks who really were essential in this project.
We do have some questions for Maggie, though. So if I can go ahead and read that out for you, Maggie, even though you can see them as well in the Q&A. You mentioned that you mentioned ancestry, but your results were based on SMP PRS. Did you apply ancestry results in your analysis?
This is a really good question, and it's really important to keep in mind. I mentioned ancestry, and then I didn't really come back to it in our analyses. So there's a lot of different ways that we kind of control for that in genetic studies. So one is that we use genetic principal components in our analysis as a control variable. And so those genetic PCs correspond really well with genetic ancestry. And so that's one thing that's like really common and it's standard practice. But there's different aspects of this question as well. So we did that. But there are different aspects of this question as well. So with the polygenic risk score, there's different approaches to that. And we actually applied different approaches as well. And our final polygenic risk score that we used in these analyses was based on a method that takes into account different ancestries, and it was based on like a genetic study of a really trans-ethnic, like very, very large population that was had a lot of diversity in terms of racial and ethnic populations. And so that's another thing that's important when you're working with these diverse populations is where are you getting your genetic information? Is it reflecting your population? And then the other aspect is that we can also do sub analyses where we focus on these different groups, on the Mexicans, on the people from Dominican Republic or from Cuba or these different places. And so we also did that. But I didn't present on it. I actually had one slide on it, but I must have taken it out. So I didn't present on that, in part because it's really hard to interpret in this circumstance because we have really different sample sizes. And so that affects our power. So that affects our ability to pick up a statistically significant result. And so we had a lot of power for Mexicans because we had a larger sample size. Actually, we had a lot of power for Puerto Ricans because we have a large sample size and a lot of asthma cases. But since that varied so much across these populations, both the sample sizes and the number of asthma cases, like, the differences that we're seeing across these populations is, we can't say, well, it's different because really it's just a different in our ability to pick it up statistically. And so we did see some different patterns. We did see stronger associations in Puerto Ricans than Dominicans versus the other populations. But I don't think we conclude like, oh, they're different than, Mexicans or Cubans just because of this issue of power. So that's something that like, it's important to study further, like have other large populations like this. This is probably the largest study of the microbiome in asthma, but we only had 191 cases of asthma. So even though we had 2,000 people, over 2,000 people, it's still relatively small sample size in terms of the cases.
So I actually now have a question, but I'm going to hold my question until after this question. So I was like looking at it like that. Do you think that the sources of microbiome samples for asthma and obesity could be different? I assume you used gut microbiome for both.
Yeah, so these samples were all collected by the GOLD cohort. And so when we say gut microbiome samples, they're actually fecal microbiome samples, but people don't want to just, keep talking about poop. So they were all collected in the same manner across participants, regardless of their phenotypes. So the samples, among obese participants and participants with asthma, they were all collected in the same way. They were all analyzed in the same way. So that's not a concern in this specific study, that there's, kind of differences in the data across participants. But when we compare our results to other studies, that is something that's important to keep in mind. With microbiome data, there's a lab effect, different labs will show up with different microbes. There's geography that's important. There's all these different factors that can influence your results. And so that's important to keep in mind as we compare across studies, that they do have different methods.
So the last question is my question. This is a Natalie question. And I'm listening to what you're saying. And as someone who grew up amongst very diverse Hispanic and Latino populations, I understand culturally and genetically how diverse those populations are, even within a particular group. So even amongst Dominicans, there's a lot of diversity.
Right.
But I can't help but feel like there's a really big like tweet length summary in terms of clinical applications of your work or implications, I should say, and then more the layperson's implications of your work. And I was hoping you could close us out on those two thoughts.
So the clinical implications of this work first. So, I mean, the clinical implications are kind of like, I think a lot of microbiome research is like not quite there yet, in terms of like actually, like, can we apply this, in practice? I think one of the thoughts is that, like, we have these polygenic risk scores, and in the clinic, they could be applied. So before someone develops asthma, you could know, like, this person's at higher risk for asthma. And we could potentially do the same thing with, the microbiome or other types of omics information. So kind of profile people before disease starts. Our results aren't directly applicable to that because this was a cross-sectional study. So we were studying people who already have asthma. And one of the complications with microbiome is that, like, disease can affect the microbiome, but also the microbiome can affect disease. When we have cross-sectional data, we can't know that, right? We can't know that these microbes caused the disease because they're at the same time as these people have asthma. And so, that's one aspect that's like not quite there yet, but that is a hope. The other thing that people really hope, well, I guess there's two things. So, like, one is that this might help kind of subtype asthma, that, these microbiome signatures might differ between different types of asthma. And so that might help clinicians treat people more effectively. And then the other thing is that, there's this hope of a kind of personalized treatment with microbiome information. And so we might be able to, sequence your specific microbiome and then know, well, if we give you this probiotic, or we give you this, suggestion for dietary changes, we expect that it would change your microbiome in this way to make more short chain fatty acids, which I just mentioned, are really potent anti-inflammatory factors. And so that's kind of the hope, is that we can put a bunch, like, genetic information, microbiome information, like different information together in order to give more personalized options for treatment. And some of those, like with the microbiome, might not be that invasive. They might be kind of more subtle than, say, like an antibiotic or like a pharmaceutical approach, right? It might just be a tweak to your diet or a tweak, something like a probiotic. So that's like the clinical application. And then in terms of the layman's conclusions, I would say that, overall, we see that the microbiome differs with asthma, and that we hope will have some benefits at some point in terms of treatment of people with asthma. We don't know exactly how it will help, but, it might help, if we can make better treatment or, predict people who will have asthma and prevent it altogether, like, that would be great. And that's the ultimate goal.